So hello, hopefully I'm audible, visible. So we will be discussing today the regarding week two's is regarding any doubt. We will be solving. We will be seeing through different concepts, and if there is any doubt or any, we'll discuss about the problems. Okay, so firstly, if you all have any doubt, you can ask, or otherwise I can. Start then. So if you have any doubt, you can just interrupt. Okay, so as you all have gone through the this week's course content for the week two, so which mostly resolved around the NAPA or the nucleic acid programmable protein arrays. So let us just look at in the basic concept of the NAPA before, so that before going into the application, or what are the advantages, or what are the how it is beneficial to identify different markers or the different protein interactions or the different things. So here, as we had discussed in the previous week, as well as what we know regarding through the different lectures, and you have come across that to understand or to read, to determine, so that functional and the structural aspects of the proteome, or to all the proteins in the cell or in the body. So you need to synthesize, you need to make all those proteins, and then you finally need to study. So synthesizing all those proteins, it's not an easy process, and all those purification, extraction, and then purification becomes quite challenging and tough for the to read, to understand about those proteins. So this NAPA, or the nucleic acid programmable protein arrays, was based on the concept that with the cell-free level, at the cell-free level, you can easily identify and understand all those. You can print all those proteins in a. You can express those proteins in a cell-free system, so that all the purification and all those process becomes, you don't need to have all those process, and then finally you can perform the different protein interactions, or the different uh, uh, protein drug interactions, or anything specific application. So what is here is that we will take the purified, you will the DNA template. So as you have uh, as we have seen in the lecture, so while doing this, you need to have those genes. So for each gene, the proteins corresponding to each gene in one particular clones, so that you can then, so the concept of the gateway cloning is used, where your gene of interest is transferred into the expression vector, and so that the expression vector can just produce in the bacterial system the proteins. So here you will need to have your DNA template. Then why you need the anti-tag antibody? So here you need the anti-tag antibody so that the protein that is expressed goes and binds to that place, where you want to, so that it gets placed in the chip. So for that you need an anti-tag antibody. Okay, and then you need your, so this is your uh, gene template, and then finally what you will do is that you will add your BSA. uh, so and BSA just for non-specific to block all the spaces. Then you will add your cell extract, which for the transcription and the translation, which contains all those factors. And then finally you will get the protein, and with particular antibodies in that place, it will get attached to. So you will get that protein gets printed in a very simple and robust way within the cell pre-expression system, which doesn't require any purification, extraction, or any kind of tagging and all. So this is just a simple way. So I think you all are clear about what are the different components that are required for the NAPAs, and how this system works, so that each protein gets purified from the DNA template and then gets printed on the chip. So this is just a basic concept which was developed, and through which different assessments are done to find out the different biomolecules or the different biomarkers that are been uh, to screen for various diseases. So this is the basic concept of the NAPA. So now the thing is that it was around more than 10 years which has happened, is that the NAPA has been into the place where different uses, it is being used for different purposes. So this has been came this review article which has told about that how it has progressed over the decade, what are the different prospects are there, and what are the different access which are there. So as I had uh, told, so this is the what are the current workflow that has been in place. Is you need one recombinant cloning, GST tag, sequencing, and high throughput DNA extraction because of the cloning purposes. So you need a clone of the particular DNA, which through which for the particular gene through which you will get the protein that you will print on the slides, and with which you would like to study it. Then your bio, and then the next step after you have come with the clones required and what you want to print, and you are ready with the purified DNA template, then you need to print those DNA onto the slides. So you need to functionalize your slides so that the printing can occur without any effect. Then you need to have its high density, and as well as you need to reproducible the printing, so that the equal amounts of DNA are printed on your slides, and whenever the expression comes, that because of the presence or absence of that particular protein, not because of the imprinting of the DNA, and which results in a high amount of protein expression, high or low amount of protein expression. So you need to have a particular robust thing in place, robust QC quality control check in place. Then you need to have those quality controls which will help you to identify which will help you to identify with your uh, amount of the protein in display, and so that you will need to express your protein, and it will find out that the proteins have been expressed in all equal amounts, so that if there is any when you will be doing and performing a particularly assay, so if there is any difference between your normal and the disease condition, so you can know that it is because, not because of the printing error, but it has been because of the actually biological difference. So then you will go with the assay development. So these three are on the basis of the printing of the chips and the quality control, and to identify all those clones and all the purified DNA template. then after the protein is expressed, you will need to perform the assay, develop the assay with serum antibody, uh, serum or secondary antibody signal detection, and then you will finally do the imaging. So you can try to identify autoantibodies from the serum, or identify any protein interaction. You can perform proteins, or you can find out any PTMs or post translation modifications, any kind of. So depending on your application, you will develop the assay, and then finally you will do the uh, after the image is acquired and you have collected the intensity level, so then you will do the perform the normalization, then the background subtraction, and then you will go on with the analysis, statistical analysis, to identify that which protein, which spots are giving you the more intensity, and what is the difference between the two groups that you are studying. So this is the major workflow that has been in place throughout, with more than uh, uh, 10 years, in line with this cloning and the self-expression systems. So now comes the major improvement, where you can see the halotype system, or the multiple tags on nanowells, or non-speed spot morphology reproducibility, then mammalian systems, expressions systems, which are giving for the cell lysate, then in vitro IVT, in vitro transfer, then improved folding, level free strategies, improved detection of interactions, new data processing tools and methods consistency. So it will helping you to have an increased heat and resolution. So this was the major thing that was going on, and these are some of the improvements that have come up into this technology, so that one can get an improved or an increased uh, high throughput result from this. So this is one of the major workflow that helps you to. And so the major thing to here understand is the major components. What are the components required? What are the advantages of it? And how the DNA is, how after the protein is being expressed, how it gets tapped, it gets into that particular position. Then the important part is the quality control, to find out that equal volume amounts of DNA has been printed on the chip and the equal amount of protein has been expressed throughout, and then the interchip reproducibility. So the spots that are being printed are in duplicates, to find out that the what is the protein after the protein is expressed, what is the variation between those two particular spots, and this is will give you your intra-chip reproducibility. And another thing which will give your, in two different chips, what is the reproducibility of those particular spots, that will give your inter-chip reproducibility. So these are quite major thing to keep in mind while performing this kind of experiments, because your printing uh, if your printing is not correct, or is there any kind of difficulty, then you will find out that there are many false positive results that you will get, so your result won't be accurate. So having a good inter-chip, and both intra-chip and inter-chip reproducibility is quite needed. So if you so it just so just uh, write here. So keep in mind these two terms, because these are very much okay. So these are very much important to for concluding any kind of going into any kind of conclusion. Yeah, so this is quite important to for understanding, and so that you can understand without for going any conclusion, you need to have those reproducibility checks, so that in between the spots as well as in two different slides it has been saved. So here what are the features? So NAPA's different features are here, that in cloning NAPA tagging is required. Why tagging is required is that because you want your protein to be placed into a particular spot after your expression, so that all the so if you are looking for a whole human proteome thing and you want to have the 20,000 spots placed within a slide, so you would require that your particular protein goes and uh, place it into that particular position. So you need to have a certain tag like anti-GST, certain tags required within the vectors protein, so that it will just go and bind with the anti-GST antibodies. Then the printing molecules is the cDNA, and when it is compared with your any commercial protein array, so their proteins are get printed, but here you will need a cDNA which can express into the particular protein. So manufacturing can be done with your contact or non-contact area. So expression system here it is cell-free, but whenever you are printing all the proteins in an array, so you need to have an expression of the cell-based system, so which will give you around bacteria, yeast or different expression systems. You need to express your proteins, then so after expressing you need to extract it out and then purify the proteins and then print it on the chips. But here you can directly print your cDNA and you will make the protein express in a cell-free manner. Then here the amount of target proteins are low yield, which is in micrograms, but in the cell-based expression systems it will be high yield. So protein display is in situ, just in time. So protein here what after you provide with the cell extract, so it will protein will get formed just before the experiment, it will be expressed. Whereas for the commercial protein arrays where you need the cell-based expression system, you will require a laborious protein purification then acquisition. So then affordable, and it is quickly, you can just print your cDNA things and just can express and it will be quickly done. So another, in commercial protein arrays, if you want to print all those in a protein array, you would require to either buy the proteins or you need to go through the rigorous purification processes. And it's, if you see the cost aspect, so it will be quite low in the NAPA arrays. So just you need to have the cloning costs, and whereas commercial protein arrays it is very much high expensive, so it will lead you to all those cloning costs. Additionally, you need to purify of the price of the printed proteins, so you will require all those things. Okay, so this is one thing. And for application based, you can have broad number, which is your open system as well as a closed system. So here, so the major things, so just think about the concepts of your NAPA and the commercial protein arrays. So that the requirement of those cumbersome cell-based expression methods, where you will require the cloning, the cells will express your proteins, then you will extract those proteins, then you will purify all those things. So to cater all those problems, only came the NAPA arrays, which helps you in just the cell-free expression system. So these are all those advantages. So the proteins, though it is in the low yield, but it will give you just in time, and it will help you to get the results and the analysis done. So now what are the different applications through which you can use the nucleic acid programmable the NAPA arrays? So you know that the detection of chemical protein acetylation. Okay, so NAPA can screen for human proteins, so 6,000 genes that undergo chemical acetylation. So you can understand that you can add chemical acetylation to the toolbox of your modified protein. So uh, study the acetylation or the acetylome profile of the full proteins of all the present in human beings. Okay, so using so here what you can see that the NAPRS can be expressed using one of the general methods, that is a single or multi-step method. So we will discuss this thing in detail in the next slides. So I think that you all have got some problems to solve as well with uh, the acetylation in the what is been reported in the manuscript. So we can go through the manuscript and we can look at how the NAPRS had been formed there, how it has been used to identify the different acetylation programs. So this is the acetylation profile of the proteins and particular proteins through which acetylation was detected. So you can detect the chemical protein acetylation modifications. So here you can screen around 6,000 genes that are, so assume that you can easily detect the PTMs and post-translation modification. So here the chemical modification of acetylation without the enzymes can be detected. Uh, so would detect would be detected through this particular method. Then discovering your protein-protein interactions. So at the proteome level, so you can just it is quite simple that you can just bring out all those proteins and then display your uh, interest of the proteins and where it will be interacting, and then you can detect it with the secondary antibody fluorophore, and then you can just report that which are the different novel protein interactions. So interactions here you can see that between the query and the immobilized target protein. So immobilized target proteins are those which have been expressed from the cDNA through the cell-free expression systems, and whereas the query proteins are your proteins of interest or one-to-one identify. So if you have discovered one protein, if you have identified one protein in a disease state, for example, in a disease state you have identified one protein to be found to be protein to be overexpressed. Now you would like to find out that this protein interacts with what other proteins in the proteome. So you will just take the clones of all the genes present and print those in the array, and after just printing in the array, what you will require is that you will spread this query protein into the particular chip, and this protein with whichever proteins it interacts or it has the interaction, so it will go and bind to all those particular proteins, and after binding to it, so then you you will place the secondary antibody into the solution, and then with the secondary antibody it will be a fluorophore tag. So the secondary antibody you would be able to detect all those particular proteins that with which it is interacting in the slide. So there you can identify the protein interactions. Then you can do is that profile your interactome networks. Okay, so all approach for, and like the same way you can do for one protein, you can do for different multiple proteins and you can identify all those. Then comes as the multiplexed reproducible arrays. So combining as many as 5 different DNA plasmids. So within one spot you can combine different DNA plasmids so it will increase your array density. Okay, so this can helps, and as we have discussed that with the mother workflow that was covered and being used for quite a number of times, there have been some improvements, which includes your using it of halotype system or the multicolored imaging. So among which one is your this one, which is the multiplex reproducible arrays, where you can combine 5 different DNA plasmids within one spot, so that you can achieve a very high density of the array. So here interactions can be detected using an anti-halotype antibody. So here you can see that it increase in protein interaction and serum spinning studies, and it has the potential to facilitate your high throughput translational research. So it increases the number of display proteins per microarray by 5-fold. So with different plasmids, if you just try to spot all those array within one spot, then it will give the number of the displayed proteins. It will be quite high. It will be around 5 times greater than what it used to be. So you can understand. So with so these are the major things that are being used in NAPA. One is you can identify different chemical protein modifications, interaction networks, and one is that you can have a multiplex, so that to increase your density of the proteins. And some of the uses in the clinical uses of the biomedical uses which are being used is that serum profiling. So these are the some of the examples that have made use of the NAPA arrays for finding for serum profiling, and has given some hints and some ideas and the concepts through which you can understand that that this can be taken as used as an early detection marker, or can be used as a prognostic marker. I think that has been discussed in detail in your lecture regarding that what are the early detection markers and all. So here you can get about the immune profiling, like serum immune profiling in women with pre-invasive cervical lesions and invasive cervical cancer. So here of 2 low risk and age 10 high risk HPV types. So valuable measuring the serologic response to HPV in cervical disease. So how human, so what, so you just try to understand their experimental plan or experimental procedure. What they have done is that they had taken the human papillomavirus proteomes, so they are genes clones, and have expressed those with two low risk and 10 high risk HPV types in one of your slide, and then what it has done, that it has tried to identify the response, the serologic response or the serum response, that how in the against the HPVs of both low risk and the high risk, what is its response or how it is being occurring. Okay, so this is, so both codon optimized and non-optimized HPV types were printed on their arrays for direct comparison of the antibody reactivity. So with the serum proteins, so with the proteins which is reacting against particular proteins of the HPVs, you can find out that those particular uh, proteins are reacting with those particular are giving any immunogenic response or not. So if you have the antibody, if the patients have the antibodies for those particular proteins, then those antibodies will interact with the proteins that you have printed on the chip, and that antibody can be detected using your secondary antibody, and then you can understand that which particular region of the proteomes of the viral proteomes are the epitopes, and from there you can understand that okay, so these are the regions which are the immunogenic regions of the virus, and it has a major implication or major uh, application is that if you can understand that which particular region of the virus or which proteome region of the virus is showing the immunogenic response in the individual, then you can design a vaccine targeting that particular epitopes so that the immune response can be higher in the. So if you design a vaccine against that particular regions, then uh, you can develop the immunity in that patient, right? So this is quite important and interesting thing to understand that what is the immune profiling against a particular virus. So the basis of this I will like to share one paper. So you can see that here with the microarray you can understand the variant, the different epitopes of a particular proteome, and then you can understand, and you can then, so you can then understand and then you can design vaccines or the against that particular region. So it is quite important, and just you can go through this to understand more. So this is two proteins or the peptides by printing on those, whereas to the number with the cell-free expression system you can just easily identify those. Then here is the screening your diagnostic performance of individual tumor associated autoantibodies and antibody panel for ovarian cancer. So this is another thing. So which are the viral mediated cancer like cervical cancer, HPV mediated cancers. So you can get to know about immune profiling against those particular viruses, or for any infectious diseases you can know the response of any individual to a particular virus or a particular bacterial load against their proteomes, how they are doing. Similarly, in the different tumors, it has been found out that some of those own proteins becomes turns into antigens, and autoantibodies are reported, gets generated against those uh, proteins only. So there you can so custom for microarrays populated with your cancer related target antigens can be done, and then you can identify the different potential circulating biomarkers. So this you can easily detect through these arrays. Okay, so they have done their sequential screenings. First 34 cases, 30 healthy controls on tumors, 740 further testing. Second, so this is an excellent example of, I think, in the lecture you have, if you have gone through the requirements of your uh, the testing on the different groups and the validating of your biomarkers. So this cannot be done on the two same cohorts, otherwise it won't give you the correct picture of those particular that the biomarkers you are claiming of for are really actually it's working or not, or it's in the disease in the control group or not. So you need to have a different control group and the different disease group through which you can and then you need to have a sum on the discovery case, and the more you need to validate on those so that you can get the actual thing. So this is how so after you identify the different markers of the group, so then you need to design your experiment, and then you need to identify. I think that has been discussed in detail in the lectures regarding how you need to validate and the type by which you can validate. Then another is with the Crohn's disease. So these are all with the real life examples and the real patients samples. These have been done and tried to identify, so that all these NAPIRs have been in use to detect. So here you can see that there are 48 CD patients and 48 healthy controls were profiled for their antibody responses against all these proteins. So you can have the full set of proteome, their DNA being printed on the plate, and then try to identify. Whereas you might have another you might have some targets that you have known and that you can print in the array, and then you can screen the patients. So here you can see that at high prevalence compared to controls on arrays were discovered immunoproteins identification of the specific autoantibodies in CD. Then you are profiling humoral autoantibody in type 1 diabetes patients. So you know that the type 1 diabetes is also kind of uh, immune disorders. So you can screen against 10,000 human proteins of the or half greater than half of the human proteome across 5 NAPIRs steps. So they have performed this thing and support the use of unbiased screening to find biomarkers. So these are the different things. So VST1G. So these are the different tags they have used. So you can ask that why the different kinds of tags were used. So different kinds of tags were used to remove any biasness that okay, so certain proteins are uh, good for the GST, anti-GST, and can is getting placed on the spot, whereas others are not getting. So if you use different tags and finally it all the in all the 5 array, it gives you the same result. So it will, you are more confident that your tag has also, so you will be able to defend that your tag has not played any role or has not incurred there is no biasness due to your tag for which you are getting a specific set of results. So you could overcome that thing also. So the major bottleneck in this kind of experiments are here to get those clones to print those particular DNA there in the slides, and so that you can, and in a very reproducible manner with a high stringent quality control system and processes, and then is to express your particular proteins and to make sure that your proteins have been in a high, in a density manner, have been expressed in a chip, and then you go for the detection and then for the statistical analysis. So these are another thing that targeted screening on 80 pre-selected proteins. So this all remains the basis of how much you would like to screen. You can screen if you are unaware of that particular disease and would like to check for all the things. For example, cancer, and also you need to print all those DNA and express all those proteins present in a particular human, and if you want to have some specific targets or in some viral proteome or some bacterial proteome, then you can also easily do that. So these are identified 9 O and 7 RA disease related autoantibody target candidates. Then your so they had combined your NAPA as well as antigen array concepts for your biomarker discovery. Then, as I told you, that it is an excellent tool for your identification of your infectious and parasitic diseases related research. So host pathogen protein interactions. So with the host protein, how with the pathogen protein interact. So you will need to have your pathogen proteome printed on your arrays and high density NAPMN arrays. Then you will give HeLa lysate based cell free expression system which will contain all your transcription and the translation factors and everything. Then it will get the sequences. Then you will have the different proteins, and then you will get the physical protein interaction screening and identification for the identification of the novel host proteins, targeting or the host ligand getting targeted with that particular set of proteins. So you will be able to do that. So it is quite beneficial for the infectious and parasitic disease research as well as your early detection using the immune profiling system. Okay. Okay, so now we will go with this one example. Okay, so we will be going through one kind of example of your non-enzymatic protein acetylation, which is detected by your NAPPA protein array. So, how can you detect your protein acetylation that can be detected through NAPPA protein arrays, which was one of the examples and have been in use. So we will be looking at that. So here, if you go and read in the abstract, the abstract tells you about the full manuscript in a gist, so that you can just understand that what manuscript is about and how you can understand what manuscript is about and what it is trying to achieve and what are the conclusions. So you can know very well that your acetylation is your post-translation modification. So it happens in the cellular organelles, but mostly it is known that it occurs within the mitochondria through acetyl-CoA transferase enzyme for different physiological processes. So here in mammalian cells, it is known that your acetyl-CoA is your primary acetyl donor, and which helps in the acetylation due to relatively high concentration located in these organelles. Now, so here what is there, that in contrast, acetylation outside of the mitochondria is thought to be mediated predominantly by your acetyl transferase enzyme. So it is through an enzyme for your acetyl transferase, but here you can see that what they have tried to look is your non-enzymatic protein acetylation. So they had here their hypothesis that proteins become acetylation without the help of an enzyme, as well as without any enzymes. They also can be acetylated. So how, if those acetylation occurs in the system, can those be detected using those NAPPA arrays? So this is the major objective of this particular work which they had initiated. So what they had done is that possibility of the non-enzymatic chemical outside than previously appreciated. So previously it was mostly told that with the help of an enzyme only acetylation can be performed, but it was not very much accepted till the time this work had been done and showed that to identify those acetylation things without any particular enzymes. So they employed your they used the apparatus to see and human proteins to identify those human proteins that have undergone chemical acetylation, and then with diverse functions, and so that can interpret the proteins with different functions and cellular localization. Okay, so this so many of those identified proteins, they had what they had identified, they resided outside of the mitochondria and have been previously demonstrated to be acetylated in vivo. Okay, so this broadly throughout the eukaryotic cells and previously appreciated suggests that these translation modifications may have more diverse roles in protein function and your pathway regulation. Okay, so this is what their hypothesis was about. That they said that the whatever is known regarding that in the mitochondria through acetyl CoA or the acetyl transferase enzyme, but outside the mitochondria also acetylation occurs in protein, and that too without the help of an enzyme, and those detection of those particular proteins which undergoes acetylation would help you in understanding the different roles it plays or in the different processes it is used. Okay, so this is if it is not enzymatically acetylated, so it will be your chemically acetylated proteins. So these proteins are chemically acetylated, whereas uh, it was thought to be acetylated through uh, what you call as enzymatically. So for this initial protocol development, so what they did is that a small set of your positive and the negative control genes. So the major thing is that there were some of those proteins that have been known that it's going to be acetylated without your enzyme or outside the mitochondria, but it was there was no such robust method to identify all those proteins, like all those proteins which are showing this kind of behavior, which are showing getting acetylated without the use of any enzymes and without uh, so to details who are also there was for example in a for example, if you think that in a disease condition certain proteins are getting auto-acetylated, but in a normal condition not, and for that reason, for those auto-acetylation is creating a chain of reactions or the processes uh, which are leading to a particular disease progression. So that detection through that method was not at all possible, whereas it became quite possible after they had employed upper technique to screen all those genes or proteins which can be identified for the acetylation. So here you can see that there are positive control genes like P300, CBP, PCS, HMOF and GSN5 were acetyl transferase enzymes. So they had shown to exhibit at least some degree of autoacetylation interactivity in vitro. So whenever you try to come up with a new technique and try to say that okay, so this technique works in helps in detection, might be acetylation or can be anything helps in your detection. So what you need to say or what you need to understand or what you need to say is that uh, so what you need to say is that okay, it so first you need to uh, prove it or need to show it that what is known, that okay, there are some genes or proteins, for example in this case for the acetylation, that some of these genes or the proteins are known to produce autoacetylation. So first with the NAPA array can you detect those? with the known proteins or genes which is having your autoacetylation, so which you have known to have autoacetylation. So first the need is to identify those particular genes or proteins uh, which has some autoacetylation, so that these are your positive control. And then so these are detected correctly. Now if another protein or genes are also detected as well with it, then you can say or show that okay, so these are also detected. So these are the novel new genes or proteins that are getting detected because of their acetylation thing. So what happened is that so these are the negative control genes P53 histone H4 that were demonstrated substrate of acetyl transferase enzymes. So these were in vitro flag tagged. So here what was used was their flag tag. So as you know that you need to tag with either GST or flag, so that it can easily uh, I didn't get immobilized at the correct place. So the plasmids were printed into the array site along with an alpha flag antibody to immobilize its translated protein to the array. So with the flag antibody, it will get the with the flag after expression of the flag protein, it will just get immobilized. Now then protein expression was initiated by addition of HeLa cell based cytosine. I think now you can visualize the procedure for NAPA, so that's why understanding the basic functionality of the NAPA, how it works, like you'll appreciate this slide and this technique where you can see that purified, so you can visualize that. So whenever you now you will be reading anything about NAPA or the procedure for the NAPA arrays or the application with which NAPA has been used, so you can, I think you can imagine and as well as visualize the thing, that how the antibodies and the DNA template and your uh, purified DNA template is being added, and after the self lysis expression system, it will give you the proteins, and then it will get bind and immobilized, and then finally will get. So here also they have done the same thing. So you can visualize the thing. So with the genes they have taken SP300, CPP, PSEF, HMO, GCN5. So these are just your positive control genes which has known to cause autoacetylation without using the enzymes, and some of the positive substrate of those particular enzymes like P53 and P4 were also taken. But the important or the concept of the NAPA which was used is here, where plasmids were printed into your array slide with the purified DNA extract along with the flag tag, flag tag, so that it can be immobilized with your flag antibody, and then your HeLa cell-based lysate, and that can give you your in vitro translation mixture. Then this expression mixture was supplemented with T7 polymerase, so it will help you to transcribe the recombinant proteins which were under the control of a T7 promoter. So since it is under your T7 promoter, so T7 polymerase will help in the transcription of that protein. So then after washing, so with all those non-bound proteins which could not be bounded by any antibody or could not be immobilized, would be taken out. Right. So after washing, the quantical pure immobilized proteins display will be displayed on the array slide. So then the major important thing is that, as you've known, so now you can visualize these things. So I just this paper gives you an importance of all those steps, and as we had discussed, and how that has been used uh, to understand. So just knowing that okay, so this can do, but here you can see that it is being used practically. So here you can see that the picogreen staining, with that you need to identify that how your protein has been expressed fully through overall with equal amount or not. So firstly, if your quality control step which goes is that your DNA has been uh, printed in equal amounts or not, and then it goes on is that whether your protein has been expressed in full amount or not. So this is your thing. So after this, so then you will see that the quality of the plate was assessed using picogreen staining, and the protein expression was caused using a secondary uh, a flag antibody. So using both picogreen staining and the secondary flag antibody, you could understand that okay, the proteins have been expressed and they are in place. So if you use the secondary flag anti-flag antibody, so it will give you the signal that okay, so that proteins have been immobilized correctly. So now, so what is that? So to determine your acetylation capability of each protein on the array, so whatever protein you have printed on the array, so now what you will do is that the displayed proteins were incubated with 14 C-labeled acetyl coenzyme A. So the NAMPA has helped you to just print those particular proteins in that slide. Now you would like to check it with the 14 C-labeled acetyl coenzyme A. So now there are two methods which were tested for the acetylation. So this is this process is a part of to understand the acetylation process and the proteins that are getting acetylated or not in the two different conditions. So the NAPA thing lies on your first part, where you will be printing the self-free expression. You will perform the quality control check, and you will check that all the proteins have been placed or not, and the tagging using flag tag or your GST tag. So this is the major thing. So now you can see that 14C acylating the protein using translation reaction or after the translation reaction was stopped. So there are two methods. One is your single method and another is your multi-step method. So in your single step method, what you will do is that 14-acetyl CO enzyme was simply added your transcription translation reaction, and after a 1 month exposure to the film, the 14C incorporation. So why this 14C incorporation was there? So that you can detect it, because with the normal carbon, with the 12C, you would not be able to detect that how much has been incorporated or how much has been into the thing. So you need to identify you give a radio labeled carbon, so that you can detect it easily. So the 14C incorporation then was quantified by the integration of the spot intensity on the film. In contrast, in the multi-step method, what was done, all attempts were made to start the radio as with a minimum of pre-existing acetylation sites. The arrays were translated non-specific acetyl inhibitor S-acetyl CoA. So in the multi-step process, so they had used one acetyl transferase inhibitor as well, as so that it can help in inhibiting all those acetyl transferase, which will help you in the acetylation. So following expression, the arrays were fully deacetylated using concentrations of the histone deacetylase HST2. So using another particular enzyme, it fully deacetylated. So then following this pre-treatment with 14C acetyl-CoA and pre-process as described above. So in one step, directly 14C labeled acetyl enzyme was added, and in another thing there were two steps which was taken. So both the acetyl transferase enzyme inhibitor and the histone deacetylase has been used to deacetylase and to find out that actually which happens without any enzyme. It happens. Then after this pre-treatment, were then incubated with 14C acetyl CO and processed as described. So for both the methods, in addition to arrays processed with full transcription or translation reaction method, process lacking the T7 polymer, which result in no recombinant protein production. So another control was taken, as which lacked your T7 polymerase in the expression mixture, which result in your no recombinant protein production. now you can understand that you have the, so you need to try to detect your both 14C acetyl CoA as well as your histone deacetylase, and also try to find out what is your as a another control where T7 polymerase is not there, so that there is no false detection. One can question about your false detection, where you will get that okay, so there is no where there is no T7 polymerase, there is no expression, so you will get the control. Yeah, so this for now use if your NAPA to screen your chemically acetylated proteins. So what has been done, so after you have with a small set of positive and your control genes, so now you are ready to go for the 6000 genes screening to find out that which are having your no enzyme chemically acetylated proteins. So these were genes from this taken and encoded into a C terminal flag for GST. So as you can see that it was encoded onto your C terminal flag or GST tag in an expression in an in vitro expression vector, and after this was split into four sets resulting in about 1500 genes printed for time. Alpha GST, alpha flag antibody for immobilization, that is were processed using the single step method with the radio level 14C acetyl CoA added directly to the protein expression reaction, same the protein reaction expression itself. So on the basis of these two methods, with both the single step method and the multi-step method, uh, find out that both methods produced the results quite similar, and it was not much needed for to go into the into the multi-step procedure. So that's why it was taken that it will go with your single step method only. So after that you can see that the four array sets, uh, three array sets were produced with the full expression mixture, and two were produced with a translation mixture lacking the T7 polymerase. After processing and exposure, the fields were scanned and the intensity of each spot was indicated. So with the incorporation of the 14th way, you will get the spot self intensity and you will try to identify for each spots. So you can see that for the conformation stage, the arrays were processed using both your single step and the multi-step procedures to allow for a cross validation of the technique. So if you want to, so they just needed to cross validate both the techniques so that there is no question on that particular technique why it was used. And then you can see that you have your 14 slides with 4 being treated with the full expression mixture and the 3 lacking the T7 polymerase. Following the routine, the arrays were processed, spot intensities were quantified and you're normalized. So then according to the Z-score with the replicates being maintained, many of the genes at the Z-score they allow for a more rational cutoff for selecting each two, and as Funnel section, 2 of the 3 spots had a Z-score greater than 1 with the third spot having a positive value. Okay, so then you can see that this narrowed their list of positive hits to 31 proteins from the single step method and 27 proteins from the multi-step method, with 11 proteins being in common for a total of 47 unique candidate genes. So for example, for 47 candidate unique genes, there were around 31 and the 27 proteins. In addition, 11 proteins were which by visual had noticeable and reproducible changes in the full array skin did not meet the initial selection criteria for conformation array printing. So you can see that here NAPA skins of acetylated proteins using your top candidates. So here, so firstly you can see that you need to have all those proteins printed. Find out that immobilized, then using your acetyl CoA enzyme directly incorporating your 14C radio labeled carbon so that you can easily go and identify any particular proteins. Whereas if you don't identify or find any particular proteins, then you need to so if you don't find any particular uh, so and then in the single step method or in the multi-step procedure, you will first use an enzyme inhibitor deacetylase, and then finally you will try to incorporate and then quantify and normalize those intensities, and you will get. So this was the experiment that was done using no enzyme chemical deacetylation. So where GST was tapped and how why the GST is, I think you are clear now. So this was all about your NAPA arrays and their function. I think you are very much clear about it. So now one major thing about your in the lecture, I think you all have gone through the lecture and have found it that there were many talks about biomarkers. So I'll just reiterate it once so that you can revisit what was told. So for the biomarkers, you need to see this biomarkers can be of many types. As you can see through the NAPA technologies, you can also immune profiling, can be your both uh, early detection biomarkers as well as prognostic biomarkers. So I think in the lecture it was well explained what are early onset detection biomarkers or what is a prognostic biomarker. So what is so early onset detection biomarkers is that you want to find out diseases. So this can be happens using your, for example, anti diseases like cancers. So there is also your risk marker, early predictive markers and all. And for your prognostic marker, for example, in infectious diseases. So if one patient will go from non-severe to severe, you can identify through prognostic markers. So these are quite important to understand, and you can know that APA technology can help you in understanding those. Then the immune profiling, identification of vaccine candidates, and everything can be happen through, you can perform through the APA technologies. So now, so for a biomarker, you need. I think you have. In the lecture, one important thing was your power analysis. So you need to perform your statistical tests before you go on to design any experiment, to identify, to understand and need to the how many samples you need to see, that what are the differences are there, how much it is your occurring. So I think it has been explained in quite detail in your lecture, so to go through. So you need to first design your experiment, then you need to with the sample number and the sample things, and then you need to or then you need to identify those particular markers, which I think there are two graphs, which is your basal level expression and which is your outside. So there is a certain zones which are, you don't know that whether this is in the disease state or in the normal state. So you need to validate in a other set of control and the diseases, so that you are very much sure that the particular region what you are getting is it doesn't belongs to that particular region, or not have your false positive. So always remember this comes like there the term of false positive. Okay, and this is very much in the case of biomarker detection and reporting any biomarkers. So people, what they get is that different so one can get different false positives, so that needs to be validated. So here check it out that, so here actual values and your predictive values. So for example, you are screening your disease patients and in the normal patients. So if it is, so you will be screening, for example, taking NAPA technology only, you have printed all those particular proteins and now trying to early screen for different antibodies. So you did a pilot mode of study with 5 patients and 5 individual controls, and you found out that certain autoantibodies and getting highly expressed or showing high intensity in the chip, certain proteins. So this in the patients with the controls. So now you have taken another cohort of patients and another cohort of healthy controls, and now you are trying to screen your patients blindly, that okay, that you will be placing the serum on the same chip, and on the basis of that particular proteins of the antibodies that you have uh, came across in your NAPA system, those, on the basis of the intensity of all those proteins, you are trying to come up with the decision on the basis of your protein map, as you have that two maps with the zone of the not clear zone, or the so you are now to predict. So if your prediction is correct, like you have predicted that this particular sample is a disease and it is a disease, so that is your true positive. Whereas you have predicted that your this is a healthy control or it is not a disease patient, and you have seen that okay, and yes, it is your disease is a healthy, and it has been predicted, you have predicted it as healthy, so it will be your uh, negative values. Similarly, if you have predicted it has a disease but it is actually not a disease and it is a control, then it will be your false positive. So that gives you the false positive, but don't get confused with the false positive. So here are two things. False positive for the protein identification, and here you have actually predicted. So here, so the difference between here is that reporting one biomarker as or identifying to a particular cohort or particular experimental error, identifying a certain biomarker is your false positive. But here false positive implies with that you have predicted it is a disease, but it is not so, and it is an healthy individual. So this is your type 1 error. So the important is to understand the type 1 error and the type 2 error. And type 2 error is it is negative, it's positive. So among the question is, implications, and you need to you should try to decrease the errors. So here it is another example. You can see that actual one, the predicted node. So here you can get a 2 negative, 2 positive, false positive, and your false negative, and the numbers, and accordingly do. So here you can know that the error sensitivity, as it has been shown, to calculate the sensitivity, true positive, false negative, accuracy. So here you will get all those formulas, and then you can also calculate the precision and the recall.
मीटिंग सेंटर है तो फिर